Okay, so welcome to the 12th ECR ICMRBS webinar. I'm sure everybody's been to many webinars now, so I'm sure you all know the rules. So I'm just going to pass straight over to our first speaker.
Yes, hello, thanks a lot. Okay, yeah, hello, my name is Jan and I work as a PhD student in Remco Sprangers Group in Regensburg in Germany. And first I'd like to thank the organizers for putting together this webinar and for inviting me. I think it's a really great webinar and it's great to contribute to it. And I will talk today about fluorine NMR, or more specifically about methods that we can use to study dynamics in biomolecules with the 19F nucleus. And as you know, molecules do display dynamics on different time scales. All molecules do. You have general dynamics such as bond vibrations. And if you have a specific biomolecule, then you're probably more interested usually in loop motions or domain motions. And NMR provides a toolkit to study these dynamics, especially in this important time range of an exchange between microseconds and seconds. There are different experiments for that. And most of these experiments focus on protons and 13C and 15N nuclei, because these are elements you have abundantly in your biomolecules itself. And yeah, this is why you use them. And in certain situations, it might be advantageous to focus on other nuclei, such as 19F, that you can specifically incorporate. And just to illustrate this, I put here the 20S half proteasome. So it's where we introduce cysteine at position 18, and then you can label it, and you have 14-fold symmetry, so that's advantageous. But in the end, you end up with a simple spectrum in 1D, and this is why you can also do analysis of biomolecular motions in a very simple manner by just analyzing 1D spectra. And there have been studies, for example, for CPMG on fluorine atoms for GPCRs, very nice studies. And we thought we would like to extend this methodology that's available for studying that. So as a model system, we take the cold shock protein from Thermotoga maritima. This has been a model system for studying folding and unfolding for more than 2 decades now. There have also been studies with fluorinated tryptophans, as we used it here, with a tryptophan at position 7 and at position 29, early paper from Ben Schuler, for example. And so this is a 2-state folder that unfolds at a melting temperature of 360 Kelvin, so very high temperature. And if you're at lower temperature, then you see that the folded protein gives rise to 2 distinct peaks, tryptophan 7 and 29, and then you get exchange broadening if you raise the temperature until you get unfolding, and you get 2 peaks of the unfolded protein here. And I want to mention that at these elevated temperatures, it's not so straightforward to measure dynamics, for example, on backbone amides, because you get exchange of the backbone amide protons with solution, and then it's difficult to record CPMG experiments, for example, there. So for our experiment, I want to introduce an element that we put in all our experiments in fluorine. If you do a simple 1-pulse experiment on fluorine, you will see that you get artifacts in your spectrum if you don't have a dedicated fluorine probe, but it also depends on your probe head a bit. And so this is the FID of such an experiment. You see that there's a huge spike at the beginning of the FID. And of course, you can just like throw away these points and back calculate them, stuff like that. People usually do that. We thought it's more elegant to do an experimental approach to that. So we take these 3 pulses here, and then you can phase cycle these 3 90-degree pulses and phase cycle these together with a receiver phase, and you get clean spectra like down here. So that's, I think, the best approach in my opinion to do that. And to start with the simplest relaxation dispersion experiment, so these are CPMGs, where you put in a CPMG pulse train, and we introduced, importantly, this A-ring sequence in the heating compensation. And if you do that, you end up with very clean CPMG spectra that you can analyze in the standard way. So in this case, we have a global unfolding process. So for both tryptophan 7 and tryptophan 29, we see relaxation dispersion. And because it's a global process, we can fit these dispersions together. We usually do that with a numerical fit procedure. So we use matrices to just calculate the dispersion profile and fit it. And in this case, we end up with an excited state population of 5% and an exchange of 2000 per second. And here, I just want to mention that these delta omegas that we extract for the tryptophan 7 and 29, these are in good agreement with what you would expect from the spectra from the folded and the unfolded protein. So this tells you that this reports on the unfolding process. So the second experiment that we set up was the on-resonance rotating frame relaxation. This is an experiment that's advantageous if you want to study faster exchange, because you can go up to 20 kilohertz for your spin lock field. And the way this experiment works is that you just do a 90-degree pulse, then you apply your spin lock, you go back to that, and we do this anti-ringing sequence again here at the end and the heating compensation. And you do that for different spin lock lengths and extract the intensities, and then you can fit the intensities with an exponential decay. And from this exponential decay, you get the this is your R1 row, like your rotating frame relaxation. And so you have to record these experiments separately because it has to be on resonance. And again, we can fit these two curves together for tryptophan 7 and 29. We fit it with an eigenvalue approach that was developed by Trott and Palmer 20 years ago, where you approximate your relaxation rate by the least negative eigenvalue of your exchange matrix. And again, we have an excited state population of around 4% and an exchange of 2000 per second, more or less. And the delta omegas are in good agreement with the CPMG experiment. So next, we wanted to set up the off-resonance R1 row experiment. This is a bit more tricky. So in addition to your spin lock power, you also have a spin lock offset that you have to take care of. And what this brings then is an effective field that is now inclined by an angle theta from your z-axis, and you have to bring a magnetization to this angle to this angle theta. There are three ways essentially to do that. You can either use hard pulses with a tip angle theta, so then your pulse length depends on your spin lock offset and the spin lock field. The pulse length depends on that. Or you could do adiabatic ramping. So the disadvantage of this adiabatic ramp is that these can be quite long, and then during the ramp you already have relaxation losses, so that's a disadvantage there. And then a third method is what I would call angle by evolution. So you do two pulses spaced by a time tau, and if you do that, you use the evolution of magnetization itself to get approximately to theta, but this only works for a certain range of theta. So this is, I think the best method here is to use the hard pulses with the tilt angle theta. And yeah, we do this in such a way that we start at Z, and then we either go back to Z, or so stay at Z, or go to minus Z for symmetry reasons, let's say. And then you apply your pulse with angle theta, you do the spin lock, and you go reverse and do the A-ring sequence and the heating compensation. And we do this for different spin lock fields from 100 Hertz to 400 Hertz. And what you get then is elevated rates at the position of your ground state at 0 offset. And you get elevated relaxation rates at the position of your excited state. In this case, it's here where the U, so unfolded state in our case. Or an equivalent representation here would be to show the R2 plus R exchange rate. So there you mainly see the unfolded state at the offset of the state. And again, we extract an excited state population of 5% and exchange rate of 2000 plus minus. And here, importantly, you do not only get the amplitude of this delta omega, but also the sign of the delta omega. So this is advantageous if you, for example, have more than one state. It's easier to see in this off-resonance kind of experiments than, for example, in a CPMG experiment if you have two excited states. So just to show you, so this is a global process and all these experiments should report on the same process, of course. And so we should be able to fit it together, and that's possible. So if you look at the curves, you see, so this is a global fit that you can fit all these data together with the same set of parameters. That was nice. And in our lab, we work a lot with larger complexes. So we wanted to apply this methodology now to real life problem, let's say. And we choose this proteasome for a start. So this 360 kilodalton complex with a 14-fold symmetry, where we introduced this cysteine at the dynamic gate in the ring annulus here. And then you can label the thiol from the cysteine with BTFA, brom trifluoroacetone. And yeah, I showed you the spectrum in the beginning, and then you can see, then you can record CPMG and on resonance R1 row data, which we did here. And an important thing to notice is that the CPMG times, so if you go to these large complexes, then your relaxation rates, of course, go high. So for example, here we are in relaxation rate of 250 Hertz. So this is, of course, a problem for your CPMG time because you have to use short times, and this restricts the frequencies with which you can sample your relaxation dispersion, right? And the on-resonance experiment is advantageous here because you essentially can sample any frequency, so you're not restricted by this time. And we just show it here by sampling a lot of these small, low frequencies here. And then you can fit it all together with a single delta omega. So we thought, okay, this should be a single process with a single delta omega, and you can fit it together and do thermodynamic analysis and so on. Last but not least, I want to show you some unpublished data from a new RNA system that we now work on. It's in collaboration with Jens Wöhnert from Frankfurt University and Jenny Vögele. And this is a riboswitch, fluorinated riboswitch. And here we chose to do a simple test experiment. So this is really like the simplest experiment you can do. You just apply a B1 field at an offset omega and then do the A-ring sequence. And then you just plot the intensity of the peak of your of interest against the offset. And if you do that, of course, at the ground state position, you get saturation of your peak. And then if you have excited states, then at these positions, you'll also get saturation. And here we recorded 3 different CES profiles at 3 different B1 fields and fit them all together. So this is real data. The error bars are quite low. It works like a charm. And you can then fit it together and extract rate populations here below 2% and quite different exchange rates, actually. So we now apply this to different systems in our lab, and this is really nice experiment as well. So all in all, why do I think you should care about fluorine NMR to study dynamics? We think that fluorine NMR can complement other approaches to do that. It's certainly not the only method one wants to use, but it complements other methods. And you can use versatile labeling. You can fluorinate the tryptophans, or you can use other fluorinated amino acids. You can introduce cysteine residues at the site you're interested in. So that's what we usually do. We know where we are interested in the dynamics and put the cysteine there. Or for nucleic acids, you can use fluorinated nucleotides. And what you obtain is simple and one-dimensional spectra. So it's very straightforward to record and to analyze. It's quite fast, so usually we can obtain complete data sets and triplicates within hours. That's a nice advantage. You can do that on many propads, so many propads nowadays can record protons and fluorines on the proton channel. And yeah, we have these different experiments that you can apply for different exchange problems. And it's applicable to large systems, and I think that's very interesting for people nowadays. Yeah, by this, I want to thank the Sprangers group for discussions and help in the lab and so on, especially Remco for having me as a PhD student, of course, and Werner, who came up with a cold-shock protein, Johanna for technical assistance, and Jens Wöhnert and Jenny Vögele for this very nice collaboration with Frankfurt. And thank you for your attention.
Thank you very much, Jan, for a great talk. Okay, so we're going to pass straight over to our next speaker, Theo.
Yes, okay. Let me share my screen. Can you see that?
Yep, looks good.
Okay, let me take the pointer. Okay, so it's a great pleasure to be able to talk to you about some of our recent data on HP40 subparents today. So thanks to the organizers for organizing this excellent seminar series. So as a way of introduction, what we really like doing in the lab is just trying using solution NMR methods to interrogate complex biological equilibria. And today I'm just going to be focusing on one of the subparent systems we're working on. And it turns out that subparents are actually pretty attractive to NMR spectroscopies because they are very dynamic systems, both in the apo state and the substrate bound state. And specifically, the system I'll be talking about today is the HP40, HP70 machine, which you can see here is almost a textbook-like representation of this system. And the cycle starts with the HP40 here, recognizing the substrate. Then it passes on to the HP70, and then HP70 uses the energy of ATP to essentially promote the correct folding of the correct folding of the client protein. So the topic, more specifically, the topic of my talk today is going to be the very initial stage here, this Y-shaped molecule, the HP40. And so what do we know about it? We know a little bit about how it looks based on crystal structure of individual domains. So here's the canonical class B DNA JB1 isoform. And here we have, in the N-terminus, we have this alpha helical J domain, which is important because it binds to the ATP domain of HP70 and promotes its activity. Then we have a long, low complexity glycine phenylalanine linker that leads to these two beta sandwich domains, which are responsible for substrate binding. And at the end, we have this dimerization helix. So all this was known from the 90s, but also around the same time, there was some puzzling data that essentially show that if you mutate residues in this long, low complexity linker region, that leads to HP70 dependent cell death. That's working. Okay, so when we started this project, instead of focusing on this canonical isoform DNA JB1, we decided to use a different HP40, which is called DNA JB6. It has the J domain in the beginning. The glycine phenylalanine linker, we only have one C-terminal domain instead of two, and we don't have the dimerization helix. And what that causes is that the protein is not a dimer anymore. It's just like it forms a pool of heterogeneous oligomers, as you can see here. And we also get another low complexity domain just before the C-terminal domain. And this protein is interesting because work from the Campinga lab in the last 5 years has identified DNA JB6 as a very potent inhibitor of protein aggregation. So in vitro, you can see here a beta aggregating on its own pretty fast. Then you add a little bit of this chaperone here, and that prolongs the time course of assembly. And in cells, you can see this puncta of fluorescence, which corresponds to aggregated proteins. But when you add the chaperone, you see this diffuse fluorescence, which is soluble, essentially soluble proteins. This anti-aggregation function seems to be encoded somehow in these two domains at the end, which we'll talk about later on. And this is implicated to various diseases, such as different myopathies, cancer, virus polymerization. So the first thing we started by doing is trying to look at the structure of this protein, which was not known at this point. So we used a construct that has the J domain, the F linker, and the C-terminal domain. Here I'm just showing the structure of the JD and part of the glycine phenylalanine linker. And what we basically see, which was surprising at this point, is that the 10 residues right in the middle of this 70 residue long unfolded, previously thought unfolded region actually forms a helix. And the helix is basically comes and docks onto the other two helices of the J domain. And this interaction essentially blocks the binding of HB70, which would otherwise bind to this purple region here. And now already from this structure, you can start seeing why mutations in this GF linker region will have such a dramatic HB70 dependent effect on cell viability. Okay. Now this protein actually has the second domain, which is this C-terminal domain, which has some bit strands. And it basically the structure is shown here. It has this unusual architecture of a single beta sheet protein, which I'll talk about in the later slides. And the NMR data, basically from the NMR data, we can tell that majority of the time, around 90%, 95% of the time, the J domain and the C-terminal domain, they're doing their own thing. They're tumbling independently in solution. But when we attach spin labels in this helix 5 here, we can see a PRE effect to the J domain, the red dots here, which we expect. But you see also large PREs for the C-terminal domain, which means that this protein kind of like transiently spends some time in the close conformation, where the C-terminal domain is in close proximity to the J domain here. So in this paper from 2019, we drew this tentative mechanism where we basically say that the HB40s can exist in an auto-inhibited form, where the helix 5 kind of blocks the binding of HB70. Then this inhibition has to be released for HB70 to come and bind, and then the substrate to get handed over and the cycle is complete. So this is important because it suggests that this HB40, HB70 cycle has to be redrawn to include this extra layer of regulation provided by the GF linker through helix 5. And if you look at the sequences of the different HB40s, the helix 5 region is kind of conserved, at least in sequences of class B. And so this might be not necessarily limited to DNA JB6, but to other HB40s. And this very nice paper from last year from Rosenbeck's lab, they kind of confirmed this mechanism by using DNA JB1. Now, the real reason why we started working on DNA JB6 is because it inhibits protein aggregation. So if we can look at this figure, for example, this is a figure from Thomas Knoll's paper where they use their kinetic approach to identify chaperones that effectively inhibit protein aggregation reactions. And you can see here that the vast majority of them kind of tend to form oligomers, heterogeneous oligomers themselves. So it looks like we have almost two competing polymerization reactions. We have here the substrate that forms these toxic aggregates through a nucleated polymerization reaction. But we also have these chaperones that are in dynamic subunit exchange within different oligomeric forms. And if it happens that this subunit exchange here is on the same time scale as the nucleation rate of this reaction, these chaperones probably can capture this aggregated species, and that's how they inhibit protein aggregation. So the point I'm trying to make here is that we kind of really have to understand what are the forces between these dynamic subunit exchange and these chaperone species in order to understand how they inhibit protein aggregation. So if we go back to our structure of the C-terminal domain, from our NMR data, we can basically see that it exists in two different conformations, which are shown here. They look pretty similar. Maybe this looks a little bit longer in this state. But if you actually look more closely to this end strand, B1 strand is a very twisted conformation here, and then it changes to a much more straight conformation in this state. You can see the backbone atoms here. This is a pretty subtle conformation change that we had to do some precise NMR experiments to pick up. You can read this in this paper. And why this is important? Well, it turns out that this state here with a twisted B1 strand is effectively a monomer. And that makes sense because this twist in the B1 strand protects this state from edge strand association. But when it exchanges to this conformation here, which is much more straight, this forms dimers. And the dimers kind of bind to large oligomeric species, which we can estimate the size to 20 to 25 subunits. The monomer can also do the same, but this pathway is always preferred here. So the hypothesis and what we believe is going on is that twisting and unwinding of the B1 strand is basically what drives the subunit change in functional DNA JB6 oligomers. And why this is important for the function, though, then we can go back to this nice paper from the Emanuelsen group a couple of years ago, where what they've done is effectively they look at the effect of different mutations in this part of the protein. And basically what they find is that if they do a couple of mutations in this part of the protein, which is the N-terminal part of B1 strand, where we see all these interesting dynamics by NMR, this knocks down the affinity of the ability of the protein to inhibit the aggregation of A-beta 42 down to 15%. So our hypothesis is that twisting and unwinding of B1 not only in terms of subunit change rate in these oligomers, but also is important for the anti-aggregation activity. So just to summarize, we've shown that this GF linker through helix 5 controls HB70 binding, and the B1 twisting and unwinding is the source of subunit change in DNA JB6 oligomers. Although we have to use some truncation constructs, and we have to basically see what's going on when we include larger parts of the protein, if this hypothesis holds on. And this the work I did when I was in Marius' group at NIH. Now I'm in the Asprey Center in the UK. And thank you for your attention.
Brilliant, thank you very much. Okay, so let's just go straight on to our final speaker, Sinem, before the breakout rooms.
Yes, thank you. Thank you so much for giving this opportunity to me. I am the PhD student and the group of Professor Tobias Madel. And in my PhD project, we are trying to understand the RGG regions in the liquid-liquid phase separation and its regulation by transportin 1. Arginine and glycine-rich regions termed as the RGG-rich regions, and they found more than 1,000 human protein, and they are mainly found in the intrinsic disordered regions, and they are important for protein-protein and protein-RNA interactions. RGG proteins play an essential role in many biological processes like transcription, splicing, DNA damage repair, and so on. And the activity of these RGG proteins are regulated by post-translational modifications. And RGG proteins are involved in many human diseases like the cancer and neurological disorders. And mutations in these regions are found in the neurodegenerative disease, and the cytosolic mislocalization of the mutant proteins in the stress granules are observed. RGG regions are important for liquid-liquid phase separation in vitro, which allows the association of membranous organelle in the cells. Membranous LLPs in the cells are important because they can provide a specific compartment in the cells, which allows the cells to regulate their reaction rates and biochemical activities. Also, under stress conditions, cells can store their RNA or protein for a short time, and this allows the cells to rapid recovery after dissipation of stress. And membranous organelles have been shown in many biological processes like storage and transcription regulation, ribosomal RNA synthesis, RNA processing splicing, and RNA storage and processing. And also the proteins more than 40 kilodaltons needs to be taken to nucleus via active transport, which are regulated by nuclear import receptors. Here we are mainly focusing on transportin 1, which can recognize their cargo via proline tyrosine nuclear localization signal. Also, it has been shown that the nuclear import of RNA binding proteins can be regulated by direct interaction with RGGs and transportin 1. It's also known that transportin 1 can block the phase separation of fuse via strong binding to PY nuclear localization signal, which allows the weak interactions towards the other regions of fuse. And proteins with RGG regions can phase separate and associated with the membranous organelles in cytosol or nucleus, depending on in the cytosol or nucleus. Also, they can be chaperoned by transportin 1. If you have a closer look at this, the sequence in the RGG regions, you will recognize they are highly variable. And according to this observation, we hypothesize that residues preceding the RGG motif determines their LLPS propensity, transportin 1 binding, and chaperoning of by transportin 1. And to check our hypothesis, first I work with the designed peptides, then I move on to I use the data coming from the first part and screen the RGG human proteome. Then in the last part, I choose the one representative RGG protein as a model system. For the peptide design part, I check the human proteome by RG repeats via a spacing from 0 to 20 and RG repeats up to 19. And we saw that more than 97% of all RG repeat proteins contain 7 or less repeats. Depending on this information, we decided to work with 7 repeat of RGG and all possible residues in the exposition. Then I checked the propensity of the phase separation and transportin 1 binding of these peptides, and I found that negatively charged residues and threonine are not prone to phase separate, and they have no affinity for transportin 1. And especially positively charged residues and the tyrosine are prone to phase separate and high affinity for transportin 1. Also, we observed that the phenylalanine are prone to form aggregates. With this information, I screened the human proteome to find the natural RGG sequence, which potentially prone to phase separate and high affinity for transportin 1. And I checked their turbidity at UV 620, which is a good indicator for phase separation. I use the constant concentration of peptides and at the increasing concentration of RNA. Here you can see the sequence with the high net charge and enriched by the aromatic residues are prone to phase separate, whereas the sequence with the less net charge and less aromatic residues cannot form the droplets. With this, we showed that aromatic residues and the high net charge are important for phase separation. To validate our finding in the cells, our collaborator from Munich performed the stress granule recruitment assays. For this, they used the MG132 to induce the stress, and cells are thermalized by the addition of digitonin. Then they add the VGA to block the nuclear pores, and they gave our FITC labeled peptides to the cells. And the stress granules are visualized by the G3BP1, and the FITC labeled peptides visualized by the EGFP channel. And here the SIRP is a positive control. We already know that it can recruit the stress granules. And when you look at the nucleolin, it's also quite prone to recruit the stress granules, and the fibrillin and LS14A slightly prone to recruit the stress granules. However, first two, they are not recruited to stress granules, which is expected because they are not prone to phase separate in vitro experiments. And with this, we showed that the aromatic residues and the positive charge also are important for recruitment to stress granules. Then to check the binding to transportin 1, I titrated the peptides with the transportin, and I did the colorimetric experiment to get the binding affinities. And you can see the sequence with the high net charge and enriched by the aromatic residues have a nanomolar affinity to transportin 1. However, the others, they hold no affinity. And we conclude that the aromatic residues and also the net charge important for the binding to transportin 1. And here you can see if the sequence are not prone to phase separate, they have no affinity for transportin 1. And if they are prone to phase separate, also they have a high affinity. So we showed that the LLPS and transportin 1 binding of RGG proteins correlate. Then I checked the chaperoning of this RGG sequence by transportin 1. For that, I used the constant concentration of peptides and RNA and at the increasing concentration of transportin 1. Here you can see at the certain concentration, transportin 1 can start to dissolve the droplets. And to check our hypothesis in more natural background, we choose the fused in sarcoma. Fused protein is a model system. Fused is the RNA binding protein and mainly localized in the nucleus. It can form and it has been shown that in many biological processes like transcription, splicing, and so on. Also, they can form the droplets and reversible aggregate in the cytoplasm. However, if fuse are mutated, this cause the cytosolic mislocalization, and these LLPs will end up with the irreversible aggregates, which are the hallmark of neurodegenerative disease, like ALS and FTD. And we also know that fuse nuclear localization of fuse is mediated by transportin 1. Transportin 1 recognizes the RGG3 PY nuclear localization of fuse and also RGG3 region. For that reason, we decided to work on that area, and I introduced the mutation in the 6 different positions. Here you can see I mutate all of them to arginine or glutamic acid. Then I checked their phase separation, and what we observed that the glutamic acid mutation is totally abolished the phase separation of the fused RGG3-PY. However, the R mutation has no significant effect on it. Then we also checked this finding in the cells with the stress granule recruitment assay. This time we used the mutated version of fused RGG3-PY with the atoll labeled, and again stress granules recognized by the G3BFP1 and the peptides recognized by visualized by the GFP channel. Here you cannot see the wild type because there is so much fluorescence coming from the arginine, but on the right side you can see the quantification of this experiment. For example, the glutamic acid mutation has a significantly less fluorescence intensity in the stress granules compared to wild type, and the arginine mutation increased the intensity in the stress granules. With that, we showed that the arginine mutations increase the recruitment of fused RGG3-PY into stress granules. However, glutamic acid mutation decreased the recruitment of fuse into stress granules. And when I checked the chaperoning of fuse RGG3-PY region by transportin 1, I found that it can slightly affect the phase separation behavior of fuse. And with that, I would like to conclude. We showed that the residues surrounding the RGGs determine the LLPS and membranous organelle association. The domains enriched by the positively charged and aromatic residues can form the droplets. However, domains enriched by the negatively charged cannot phase separate. And the net charge and hydrophobicity, the key determinants for the affinity of transportin 1. And when transportin 1 bind to these droplets, the proteins become soluble. However, transportin 1 cannot bind to these domains enriched by negatively charged residues. And with this, LLPS and the transportin 1 binding correlates, and that suggests the strength of the chaperoning is essential to regulate the LLPS in vivo. And I would like to thank the group, my colleagues and my thesis committee and our collaborator, and thank you for your attention.